My pleasure to introduce Professor Yang, who joined the UJA last semester and as a cluster head of ecology, mathematics, and statistics, but he doesn't have any commitment to IOB or ecology. So this seminar also serves for his application for the adjunct faculty position with IOB. So if you're a student and looking for lab, you may want to talk to him. His major research area is infectious disease. He is a statistician modeling for infectious disease. As you know, to understand infectious disease, we need the knowledge in ecology, population genetics, molecular genetics to understand evolution of pathogens, and mathematics, as well as statistics. So it's very hard to find an expert in all of those areas. And here we go, Professor Yang. And so if you're interested in research in infectious disease, you probably want to talk to him. He has a lot of grants. Okay. He already published over 100 papers. And this is, believe me, this is just the first page of his CV. So I cannot print out all the CVs. It's 19 pages. So he, let's see. Sorry, I'm aging, so I cannot see that. So he got his PhD in 2004 from Emory, and then he did his postdoc at Harvard, right? So for the following year, and then he worked at Fred Hutchinson Cancer Center, and then he moved to University of Florida, and last semester he joined UJA. So today he's gonna talk about epidemiology and ecology of Middle East respiratory syndrome coronavirus. Okay.
Okay, good. Thank you, Liang, for this nice introduction and over compliment. I'm not expert in everything, and I mainly specialize in modeling of infectious disease transmission, and just started working with Dr. Liu on how to combine the epidemiological part and the phylogenetic part into the same model, so that we can better model the dynamics of transmission. Today, I'm going to talk about the epidemiology and the ecology of MERS-CoV. If you read the flyer before, I changed my title a little bit. I used to have transmission as well, but I found myself too many. I have to cut some, so I moved that one out. So today, we'll only be talking about the epidemiology and the ecology. Hope you will be interested in these topics. Right. So this is joint work with Dr. Li Chunfang and Wen Changshi at the Beijing Institute of Microbiology and Epidemiology, as well as Dr. Anna Zhang at Shandong University. And I was a visiting student in my lab at UF. That's when she was leading this project, part of this project. Please feel free to stop me anytime if you have any question. Sometimes I can only use that mouse. Right. So some background about the disease and the pathogen as well. You're probably quite familiar with this disease because of the pandemic of SARS-CoV-2. MERS is a respiratory infectious disease first found in the Kingdom of Saudi Arabia in September 2012. As of January this year, there were a total of 27 countries that has reported about 2,600 lab-confirmed cases, together with 935 deaths. So that's a pretty high case fatality rate. We use the CFR of about 36%. Luckily, human-to-human transmission is pretty rare, and many occurred in healthcare settings and households. And in healthcare settings, it's pretty much patients who are immune compromised. So that's why they are at high risk of person-to-person transmission. And the zoonotic infection is mainly driven by exposure to the dormitories via droplets, fomites, and consumption of some raw products of this animal, such as milk. And people also suspect the use of camel urine as a traditional medicine is another transmission route. And given that has been no effective treatment or vaccine yet, so people are very concerned about the potential of this virus causing another global pandemic if it's fully adapting itself to the human population. And in 2018, WHO incorporated MERS into this, its research and development blueprint, which lists a variety of disease pathogens that people will, spend more time in developing vaccines toward those pathogens. And about the virus itself, it's RNA virus with a genome about 30 kb long. MERS-CoV belongs to the lineage C of the genus beta coronavirus. The famous or infamous SARS-CoV and SARS-CoV-2, they belong to lineage B. And the common cold coronaviruses, OC43 and HKU1, they belong to lineage A. So they're related, but a little bit distant from each other. MERS-CoV was phylogenetically classified into clade A and clade B. A includes the early detected human cases, viruses, and B includes the more contemporary viruses. But however, this classification was based on pretty few number of sequences. So we've done a much more comprehensive work on this, and we'll show that later. Although the disease was quite new, but circulation of MERS-like coronaviruses in dromedaries in Africa and the Arabian Peninsula occurred well before 2012. And a little bit about the immunity background of this virus. The virus uses the S1 protein to bind to two types of host cell surface molecules. One is called the DPP4, and the other is the well-known alpha-2 or alpha-3 static acids. And it replicates mainly in the nasal I may not have the correct pronunciation, please forgive me for that, epithelium of dromedaries, but only in the lower respiratory tract of humans. So that's probably why it's so highly transmissible among the dromedaries, but not so much among human beings. All right, so that's enough for the background. Let me get into the meat. So we'll try to identify determinants for case fatality rate and also characterize the spatial division pattern of the disease. And we want to also look at evolution and migration history of the virus. Finally, we'll provide some more comprehensive picture about the ecology of the virus and associated social environmental drivers. So this talk has 2 parts. The first two are part 1, and this is part 2. Okay. So in this slide, I talk about the data sources. We first collected data on all confirmed human cases, and there are 3 major sources for that. We looked at the official reports of WHO. Now, WHO does not provide the line list it used to. So we were lucky. At that time, we got a line list. And the Food and Agriculture Organization of the United Nations, as well as the health departments of affected countries. So we collect the demographic data whenever it's available, and the dates of critical events like onset, hospitalization, and lab confirmation. The most important one is exposure history, but it's very simple. It's just whether the case has had a history of exposure to animal products, not production, sorry, and or other confirmed MERS patient. And we validate this data via literature search. We also collected a bunch of social environmental variables, which are listed here, and we'll utilize them, especially using, for example, meteorological data and land cover data for ecology study. The gene sequence data were downloaded from gene bank, and we use data up to the June of 2020, and that's also the time upper limit that for the case data we downloaded. This is an epidemic curve actually provided in the WHO report, and it only listed the cases in the Eastern Mediterranean region. It's not for the whole world, but because we want to check the seasonality, so we'd better to focus on the endemic zoonotic region instead of the local region. And it covers from June 2012 to January 2023. This is more comprehensive than the data that we use. We use up to June of 2020, but luckily there weren't many cases after that. I mentioned it was first discovered in September 2012, but you can see here, actually the earliest case showing here can be traced back to early 2012. So there could be some cases retrospectively identified. All right. Majority of these cases from Saudi Arabia. That's understandable. Everyone knows that. And we see a huge peak in 2014 and some subsequent large outbreaks in 2015. And you can also see there were about 2 peaks per year and sometimes 3 peaks. Right? One in the early spring and the other one probably in the late summer or early fall. And the early ones in the spring, it's more like animal exposure in triggered spillover. So here is a summary table of the kind of baseline characteristics of all the MERS cases we included in our study covering this period. Right? And I colored some of these interesting results. In Saudi Arabia, it has far more number of cases as well as number of deaths compared to other countries. And if you look at the CFR, it's also much higher than other regions. In South Korea, I should say South Korea, sorry for that. The number of female patients were much higher than other countries. That's possibly due to, because in Korea, if you know the history of MERS, there was a big outbreak in, if I remember correctly, it was in 2015. It's kind of driven by one single imported case, and that patient kind of stimulated large outbreaks in several hospitals. Right? So it's purely kind of a person-to-person transmission, and in that case, it doesn't really distinguish between different sexes, right? But for animal exposure, we'll see later, most of the patients with animal exposure are men. And so that will make some difference. And in these countries, KSA, UAE, and some other countries, the majority of patients are male. And South Korea also has a very large percent of hospital-related patients, and that's exactly because we only had hospital outbreaks in that country. And the time from disease onset to diagnosis was shortest in South Korea. And that's also understandable because those were hospital-triggered investigations. They are very efficient, right? In the United Arab Emirates, we noticed that the age was, the patients were much younger compared to other countries. And that is probably because of the high proportion of healthcare workers compared to other countries. And this country also has the highest proportion of asymptomatic infection. How you identify asymptomatic infection is a very tricky question. Usually, if for syndrome-based surveillance, you only can find symptomatic cases. Because of this high proportion of healthcare workers, probably they did some regular monitoring of infection status, and that's how they identified asymptomatic infections. And this country also has a very long time from disease onset to death. The reason for that is unknown, but probably related to the management of patient or facility in the hospitals. So that's for the summary table. Any questions? So that's the basic epidemiology. Let's go to more interesting materials. In this table, we compare the characteristics between cases with animal contact and those without. About 1/4 of patients were reported animal contact. So we can see patients with animal contact were much less likely to be female, right, compared to this proportion. And they were older. And the case fatality rate was much higher. But we need to be very careful for interpreting this much higher case fatality rate. Because this animal contact was later, it's confounded with other factors like age, like baseline chronic conditions. Right? If we really want to assess animal contact effect on case fatality rate, we need to adjust for those confounders. It's also less likely to be asymptomatic, but that again is a question mark. That probably does not give you any epidemiological. It doesn't describe epidemiology in that regard faithfully, because for people with animal contacts, many of them probably asymptomatic and they were not captured by the surveillance system. And they had much higher proportion of underlying conditions, as I mentioned before. So they're probably at high risk to death. And they generally had a much longer time from disease onset to diagnosis. So they were found later. So that will also probably increase the fatality rate. But for some reason, it has slightly longer time from disease onset to death, which we don't quite understand. Probably related to some immunology issues. If you look at the proportion of animal contact, the patient with animal contact across the years, now these proportions are the low proportions. Even though we had fewer and fewer cases over the years, we actually see a different story for this proportion of animal contact. It increases up to 2018 and then kind of plateaued. And it's not decreasing yet. I'm not sure about the most recent years. Data are not available yet. Okay, so this figure shows the spatial distribution of human MERS cases in the whole globe. We categorize the countries into 4 categories. The first 2 categories corresponds to the colors of, I would say, maybe I can call that pink and a little bit of purple. Those countries had zoonotic transmissions, basically animal exposure. The pink ones reported a human-to-human transmission, and the purple one did not report, or at least did not find concrete evidence for human-to-human transmission. And if you look at this map, only Kuwait is the country with colored with purple. And it doesn't really mean it did not have human-to-human transmission. I'll explain why. The other two categories are the countries with with mainly imported infections. So we're colored with yellow or brown. The yellow indicates there was human-to-human transmission following the importation, and the brown ones, I believe, are the ones without subsequent human-to-human. Oh no, sorry, it's reversed. The yellow ones did not have human-to-human transmission, but the brown ones had this type of incidents. Right. And on each country, with reported cases, we impose a bar chart like this, and the size of the bar indicates number of cases, and then the colors indicate animal contact history. The red indicates the proportion of patients with animal contact. And if you look at Kuwait, it's basically red and green. Green simply means unknown. Right. So whether there were human-to-human transmission, we don't know, just not confirmed. Okay. And interestingly, Qatar is the country with the highest proportion of animal contact, followed by KSA, UAE, and Oman. Some small countries like these Bahrain has too few cases, so I wouldn't make any conclusion for that. Some European countries also reported the patients with animal contact, as well as here in Southeast Asia. But these people most likely already had animal contact before they entered those countries. So to look at what factors contribute to death, the risk of death. So we performed a standard logistic regression. For all the variables that we have that we think are related, we throw them into the model. We perform the univariable analysis and the multivariable analysis. And all these factors we throw in are significant in both univariable and multivariable analysis. These are strong predictors for case fatality. Let's just go ahead with the multivariable analysis. Don't look at the age, sex, and animal contact yet, because we have interaction terms in the model. And these numbers, like this high number, 10.52, does not represent a marginal effect of the age group. It does not. It just specifically to one particular subgroup, and I'll talk about that later. Right. So let's just look at the effects of, say, a healthcare worker has much lower risk of death, and underlying condition. Well, that's a more than 3-fold increase in the risk of death. OTC stands for the time from onset to confirmation. It's basically diagnosis, right? Then this has also significant results. In a longer time from onset to confirmation increase the risk of death. And if we compare the years, we divided the time into 3 segments. And this early phase, this middle phase with large outbreaks, and this later phase with very few cases. And all compared to the early years, we can see a jump in the risk of death, that's probably, at least partly due to the constrained, exhausted healthcare facilities because of the large number of patients. And in the later years, we see a slight decrease in the risk of death, possibly related to improved patient management and also the much fewer number of patients. So we left out the most three interesting factors: age, sex, and animal contact. So let's get into those. In this table, we look at age effect, conditioning on sex and animal contact. Let's focus on the adjusted odds ratio directly. Right? These are all pretty big numbers, all significant. But let's do some comparison. Let's first compare the results between female and male, right? But you have to condition on the same contact history, right? So with contact history, with animal contact history, if you compare female with male, and then without contact history, female with male, right? Without actually compare this guy with that guy. So basically, this is bigger than that, and this is bigger than that. So within females, age effect is more prominent, regardless of animal contact history. Age effect is basically comparing the older age to the younger age. And this number shows in the previous table. It's just for this particular subset of population. So age is the most prominent driver for the risk of death. And that's not affected by either sex or no, it is affected by sex. Right? In female, it's more prominent. And it's not that affected, it is also affected by animal contact. Let's compare the animal contact history. Okay, so if you look at just compare with to without, and with to without, right? You can see without animal contact, age effect is more prominent, regardless of the underlying sex. So that's quite interesting. Why is that? Why is that? Remember, males, many of them were actually farm workers. They had animal exposure. So we can reasonably speculate that these patients were constantly challenged by the virus. Okay, probably there's a buildup of immunity towards this virus. So after adjusting for the underlying conditions and adjust for the adjust for underlying conditions and some other risk factors, we can see the difference in the age group between people without animal contact. With animal contact, the age effect is not that prominent, but without, it is very prominent. And most of animal workers, they were older than younger. So that's for age effect. Let's look at the effect of gender. So I have to, effect of gender. We don't see many strong signals. The only statistically significant results is within the subgroup of the older patients without animal contact. And the male has a much higher risk of death than female. However, we also notice that this effect is have some kind of a marginal signal here. And it is in the opposite direction compared to what we see here. And this subgroup is for the for the older patients with animal contact. So that's also quite interesting. For younger patients without animal contact, male has much higher risk of death, but within older patients with animal contact, male have actually lower risk of death. Then that again gives us a difficult question to answer. Why is that? However, we should also notice that within the category of older patients with animal contact and female, we have small number. Right? So that's why this is not statistically significant. So that could be just due to random noise. So maybe we should not overinterpret this 0.45. Maybe I should use a mouse because I don't think. Can they see my laser point? Means online.
No.
They cannot see that. Maybe I should use mouse. Yeah, let me use mouse. I apologize to the online audience. All right. Now, finally, let's look at the effect of animal contact, conditioning on sex and age group. Now here, let's just focus on the significant results. The most significant results is this guy. Let's ignore the unknown category, just to focus on with versus without, right? So within the subgroup of female patients and younger female patients, we see a huge increased risk of death for patients with animal contact. Remember, these are female patients. Most animal workers were males. So females, if they had animal contact, it's probably more likely to be occasional contact, not a professional contact. So that means you may not have the built-up immunity against this virus. Right. So once you had contact with an animal and got infected, then it's probably a high risk of death. And this virus just jumped from animal to human, has not been adapted much to the human immune system. But if you look at the subgroup of male patients, the older male patients, if you look at without animal contact, you see a huge reduction in the risk of death. And most of these male patients with animal contact, especially the older ones, they were actually animal workers. They had probably long exposure to the virus. Any question? A lot of questions, information here. Difficult to digest in such a short time. Oh, I already see people have put in chat. Let's see.
I think that's from the setup.
Okay, let's move on. So those were for the risk factors for death. Now let's look at the spatial diffusion pattern. So what we did here is we look at the time from the first case overall to the first case in each local district. Right? That's the first time the local district was invaded by this virus. Okay, with this time as my outcome, I did some, spatial smoothing to construct this contour plot. Right, and this contour plot can tell me the diffusion pattern. And the wider gap between the two contours indicates let me put it this way, because the gap between each contour plot is kind of fixed for the time interval, 200 days. And a wider gap means a longer spatial distance. So that means a faster spread, right? The first identified case was here in a city called Bisha, somewhere in western KSA. And this star marks the first patient that sparked a outbreak, right? Human patient cluster. So this Bisha is kind of in the central point of these contour plots. And in the more recent times, we can see actually faster spreading, and the direction was towards Oman and somewhere of UAE, right? But there's a caveat of this plot. We did not show the uncertainty, right? So probably here you have fewer data, then uncertainty could be high, and whether the conclusion that they have faster spreading may be just a noise. Right? But however, if you look at this figure, this map here, let me use mouse again. If you look at the map in the right panel, you can see in this corner, we still have a bunch of cases. So those cases will inform the construction of this contour plot. So the signal here in this corner should be reliable. Right? And you can also notice that we overlay this map with the transportation network. The white-black lines indicate railway, and these gray lines indicate the major roads. Right? And you can see a pretty good overlap of the cases with this transportation network. And that's what we're gonna show in another model fitting. So here we performed a survival analysis. Again, the outcome is a time from the first ever reported MERS case to the first importation of case into each district. The so-called district here, it is a second level administrative unit in most countries except for KSA, because KSA is relatively big. So in case A, we use county, which is a third level administrative area. And we fit this survival model, trying to look at, this is not an ecological model. It's just to try to look at what might have contributed to the invasion of the virus into these different spatial units, right? Again, let's just look at the multivariable analysis. And here we are estimating hazard ratio. The most prominent hazard ratio is related to the intersection with the main road. After that, it was elevation. The higher elevation, the higher hazard, higher ratio. And also intersection with railway, it more than double the hazard of invasion. And interestingly, the coverage of cropland, it seems to deter the spread, which is also understandable, right? These bio variables, they are meteorological variables, and I listed the interpretations here. Bio 1 simply the annual average temperature, and the bio 2 is the average diurnal range of temperature, which is simply is kind of the average the difference between the maximum temperature and a minimum temperature within each month, then you take average across all the months. It reflects the variation over time, but within each month. So these two had shown some significant results, increasing the higher temperature or the higher mortality increase the hazard of invasion. Finally, comes down to the something that you may be interested in. So we collected the whole genome sequence from gene bank, and we analyzed these sequences using the toolkits provided by NextGen. The sequences were aligned using MAFFT, what do you call it, math?
Yes.
MAFFT, math, right, math. Okay. And the alignment was trimmed to a reference genome in gene bank. And we constructed the phylogenetic tree using the maximum likelihood approach implemented in the IQ tree software. And for the final geographic analysis, we use the tree time to estimate the location and host type of those internal nerves. An initial analysis shown on the left-hand panel suggested that the sequences from bats are denoted by these pink leaf nodes, and hedgehog denoted by these yellow leaf nodes. They separated, kind of distant from the main clades of sequences formed by human and camels, as well as a single strain from Lama Kalama, I think, the animal. Right? It is hiding here. Okay. And then we just exclude all the sequences of bats and hedgehogs, and only focus on the human and camel genes. Right. So on the right-hand panel, you can see we had several clusters for human and camel genes. We have 5 clusters. The largest cluster is called C5, which contains the majority of the sequences. And you can see the mixture of human and camel sequences throughout the tree, except for the most recent ones, where I think it was more like a sampling issue rather than a systematic pattern. So this mixture of human camel sequences suggests that there were probably constant importation or spillover from camel to human. Now, this figure shows the phylogeny in a much better way because we can denote the time here, right? So although the human and the camel sequences are mixed together, but if you look at the samples collected after 2016, the human genes are quite distant from the camel genes, right? And the common ancestor was dated around 2007. And the posterior probability for the host being camel, human, kind of 50-50. Related to the sensitivity analysis, we downsample the human sequences because there's just too many of them. And then the camel will have a 99% for the posterior probability being the common ancestor. We also try to associate the case fatality rate with sequences. So what we did is we kind of divide the spatial temporal range into several chunks and calculate the case fatality rate within each chunk. And then we look at the individual sequences and associate them with the case fatality rate in that chunk. All right, the location and the time. And then we for each sequence, we have a bar with a color indicating the underlying case fatality rate in that space time chunk. And then we average for each cluster to compare the potential difference in case fatality rate between clusters. If you compare C5 to the other clusters C1 to C4, you cannot really see much difference between the two. Actually, there's only 1% difference in case fatality rate there, only 1%. But it is significant, probably because of the sample size, but we cannot overinterpret that. If you compare only the subclade C5.1 with C1 to C4, we actually see a 4% difference, which is not true, right? So that means the more recent clade is probably more lethal. We also associated the incidence rate with the sequences, but we cannot see much signal there, so I'm going to ignore that one. Oh, there's another thing I want to show. Okay, so we remember we performed some phylogeographic analysis. So let's take a look at the movie. Hopefully this movie will show. Let's see. Just copy paste. Oh, we're not. Maybe I should go back to the original mode and just copy paste this one. See if we can do it. I'll stop sharing. Share this. Share this. See if it works or not. That's strange. I thought it's a E there. So. Yeah, it works now. Okay. Now let me. Oh, I have to move it to.
Okay. One more time.
Oh, I have to stop sharing.
Again.
Oh, I see. I can lost it. Yeah, okay. Right. That's fine. Okay. All right. Now, let me click play here. That should have been another. This only shows this. I want to see that geographic location.
I'm scrolling down, maybe. Scrolling down.
Okay. All right. Yes. And then replay it. I'm sorry for this problem, but it's kind of out of my control.
Play.
Where is play?
Just click play.
Here. Okay. Yep. Now let's take a look. almost finished. All right, good. Okay. Now let me finish. Oh, a skill. That's correct. To share the screen, right? Okay. Right. Okay. Now some highlights for that movie. Right. The top three most likely locations for the common ancestor as a route were three cities: Riyadh of KSA, and the Nile Delta region, which is in Egypt and Jordan. The posterior probability is 31, 17, and 12% respectively. And Riyadh appears to be the major source of exporting infections, both locally and internationally. And it is the common ancestor node of subclades C3, C4, and C5 for 99% of time. Okay. And the early exportation to Egypt and Jordan occurred well before 2010. And the circulation of the MERS-CoV among camels in East Africa possibly started also before 2010. The virus migrated from Egypt to Ethiopia during 2011-2013, and subsequently to Kenya during 2014-17. And that's partially supported by a serological study conducted in Egypt in 2013, where they also sampled the animals from Kenya and found nothing. The intense migration from Riyadh towards local cities in KSA, as well as up to Abu Dhabi in UAE, started around 2012, which matches our knowledge. And Abu Dhabi joined Riyadh as the second hub for exporting the virus, both locally and internationally. So that's quite early. And the model also captures the opportunistic exportation to US as well as to Asia. So we also did some kind of detection of positive selection across proteins and the sites. Remember in the previous plot, see here. Now I notated here branch A and branch B. Branch A is kind of separating the hedgehog sequences from the rest, and branch B is separating hedgehog and bat sequences from a human and camel sequences. Right. So we performed the session the positive section analysis along these two branches. Okay. The first three rows are for branch A. We identified three proteins, the non-structure 3 nucleoprotein, and this 1AB polyprotein is, I believe it's from the open reading form 1. I may be wrong, but that's my impression. And we identified the signs with positive section only in this 1AB polyprotein, not in the other two. Along the branch B, we identified these two proteins, including the spike protein. Right? For the branch A, we did not detect spike protein for any positive section. And also for the spike protein, we identified these sites to be under high pressure for positive section. And the three ones marked by red, those are newly found. They did not appear in the literature. And we all know spike protein is the one that's most interactive with animal cells, right? How much time do I have? It's
How
already,
much? Yeah, already.
okay. Then I'll probably ignore the ecology part. For the ecology part, I can just mention some main findings. What we did here was we used several machine learning approaches like boosted regression tree, supported vector machine, and random forest to learn the ecology of this virus. And then we build a meta learner on top of the three to give us another round of prediction. And turns out that the ensemble machine, which is a meta learner, outperforms the three base models. Not by much, but a little bit. If you look at the AUC, if you look at the accuracy, as well as F1 score and kappa, not really a good performance in terms of sensitivity, but sensitivity here is a little bit deceptive because we have very few positive cases. This model is based on about 100 positive districts with positive with confirmed cases, and about 400 districts without any detection. And we did identify, we did identify the most prominent risk driver for ecology of MERS-CoV is actually something called the coverage of bare land. Bare land, that's very difficult to define. Basically, the land without many, vegetation, trees like that, right? But we also adjust for camel density here. Camel density is picked up by the model, but not as a very prominent driver. So we want to control for camel density. Then it's bare land. In addition to camel, it may also contain other animal hosts that we have not discovered yet. Okay, so let me just draw some conclusions on the epi part and the viral evolution. So we observed that the cases with animal contact tend to be older, more likely to be male, symptomatic, and also having underlying conditions. Even though by our logistic model with interactions, we kind of see that once you're adjusting for underlying condition, adjusting for age and gender, animal contact actually does not always increase the risk of death. Remember, in the older population and the male, it actually decreases the risk of death, right? But overall, for those older males who are farm workers, animal workers, because they had underlying conditions and they were old, overall they do have a higher risk of death, and we need to pay attention to that. So we should consider, promoting some preventive measures like educational campaign or personal protection equipment for those workers. And animal to human transmission events mainly occurred between January and March. And animal and human to human transmission occurred later in summer. So that suggests the importance of blocking the spillover from animal to human in the early spring. And also mentioned, we downsampled the sequences after 2014, many human sequences, and then camel becomes the host for the root ancestor with a very high possibility. And we also found Abu Dhabi of UAE is a hub for international exportation of this virus. So this virus probably we should recommend some kind of screening procedure for infected travelers. And the novel amino acids positions that we found on the spike protein associated with positive selection, those can be potential targets for development for future antivirals and vaccines against this virus. So that's all for that material. The API paper is published in this journal, and the Ecology paper is published in this journal. Thank you very much. Any question?
Look has a question.
Yes, please.
I'm really interested about the phylogeny. Could you go back to the figure 4 or 5?
Yes.
Yeah, that's the one. So since after 2016, it's really interesting, like human MERS and camel MERS are pretty much distinct. Do you think it's because the sampling bias of the camel, or do you believe there's somehow a low level circulation of human population MERS? Yeah.
That's a good question. I don't have the answer. My intuition is sampling bias is at least a contributing factor. It's hard to imagine because this virus is endemic in camels. It's hard to imagine it's gone. I don't think it's gone.
So Brian in the chat would like to know what type of models make up your ensemble.
Oh, so the base models are a random forest, full-state regression tree, and supported vector machine. And then on top of that, the meta learner is actually XGBoost. Does that answer your question?
He said, thank you.
Okay. Yes.
So it seems like this is a fairly small tree. Why did you end up choosing next strain, like using the next strain pipeline as opposed to a different tree building pipeline?
Was not my decision. My intuition would always try maybe like a beast first. However, I wouldn't say it's a small data set either, because we have, I think, about 500 sequences together. Beast mainly sometimes we want.
Yeah.
And another nice thing about the next strain is that it provides, all sorts of tools together for you to use, and with this nice movie, right? So I think that's probably the only reason why we chose the next strain. But I think definitely you can try Beast as well, especially for the geographic analysis. Be more interesting. Yeah.
Could you tell us more about surveillance or genome sequencing of camel or like reservoir species? I think that's really important because can give us a good background about circulation in these reservoirs and the spillover between.
Yeah.
Camels.
I'm not quite sure whether there is a systematic surveillance system for animal. Actually, in terms of the animal host, although at least the bats and hedgehog here, if you look into the literature, it's still kind of debatable, right? We all have this common sense. Bats is the natural reservoir for all types of coronaviruses, including MERS-CoV. But as a matter of fact, so far people have only found, to my knowledge, kind of similar gene segments in the virus as lived from plants. That's not considered as a very solid evidence by many biologists and immunologists. Yeah, I know less about the hedgehogs, so I cannot say much about that. So the primary animal host that's solidly convinced people is camel, drumlins. And we should do surveillance. Yeah.
I have a question about student training because you're applying for adjunct faculty position here. So what type of students would you expect working in your lab, or how would you train the bioinformatics students?
In terms of bioinformatics itself, I need partner like Dr. Liu. Myself, no, I cannot train you in that regard. I'm very interested in this field, but myself, I need to be trained as well. But my vision is that, you know, we don't have this vision. We want to combine epidemiological data, gene sequence data. So far, we only talk about the pathogen sequence, right? It could be human sequence as well, but that's a long shot. And also like human movement data all together to inform us about the transmission dynamics. And that's also very important to understand how the virus is gonna evolve in terms of its transmissibility, pathogenicity, and so on. Right. So far, there have been some efforts in this kind of joint modeling in the past 10 years, but it's still under investigated. It's still a very promising direction. So anybody with interest in both bioinformatics and statistics and computational methods are welcome to join this effort. Yeah, sure.
It says elevation was insignificant in the univariable analysis. Why was it included in the multivariable and ended up being?
A good question. Let me check.
That's from Bohan.
Elevation, that's for the survival analysis, is that? Let's see.
Yes.
Yeah. This is a kind of more like a statistical philosophy question, right? You can, because here we did not explore the potential interactions explicitly, right? So there could be interactions among these factors. And in the univariable analysis, it's not significant. That's actually the p-value is very close to 1, but that does not mean it's not having any interaction, right? Or it could be a confounder, because if you look at the coverage of a bare land with elevation, there is some correlation there, right? Not very high, not super high, but there is some substantial correlation there. So we cannot just rule it out. So in the multivariable model, we did not just leave out those not significant in the multivariable model. We actually add them back one by one to check whether they're going to become significant after adjusting for other factors. So that's the reason. Thank you.
All right, if there's no questions, let's thank Professor Yang. That's it, right?
Yes.
Okay.
Yeah, I think I set it up for, I know I have to clean up, so I will be there.
Yeah.
I think that's sending us week. Yeah.
How do I sign up? Should I go?
You can
look?
just go.
You know what? You just show
I'll
up.
show up by the way.
Have a booth, I guess.
Okay.
It normally